So today we'll start a new topic from industrial pharmacy because we have started our physical pharmacy. We have almost completed our pharmaceutical engineering. So few topics are there from industrial pharmacy which are not dependent on your chapter from physical pharmacy. So that's why we are taking pre-formulation first, and in the on the 15th of February. So I will take a surface tension interfacial phenomenon. So pre-formulation again very important chapter from your from your industrial pharmacy. So industrial pharmacy start with this chapter pre-formulation. So at the as the name indicate, what do you mean by pre-formulation? So pre means before. This term pre is the for before, and then next one is a formulation. So study, whatever analysis, whatever determination is done before formulating something, before formulation of dosage form, before designing the dosage form. So whatever study is there before the formulation of dosage form, that is known as a pre-formulation. Now what kind of study you have to do before formulation? Suppose it will take around 20 to 25 years to develop a new drug. It will take around 20 to 25 to develop a new drug. Once new drug has been developed, so and once that drug has promised pharmacological effectiveness in the animals. So वो ड्रग अगर प्री क्लिनिकल ट्रायल्स में सक्सेसफुल होता है, if the, if that drug is promised the effect in a preclinical studies, then you have to start your pre-formulation studies also simultaneously, and simultaneously you have to do your clinical trials. Okay, because once it is clinical, once the drug is past clinical trials, then uh, you you are in a hurry to market that product. So that's why you will get a very less time to study the pre-formulation parameter or to study the physicochemical properties of the drug, right? So around it will take around 20-25 years or 30 years to develop a new drug. Okay, so drug discovery, it will take around 30 years. Okay, so during the drug discovery, these are the different phases of drug discovery: medicinal chemistry and pharmacological study, then pre-formulation and research, formulation development, process development, then analytical and research development, and then lastly toxicology and its metabolic study. Now drug discovery, it is done first. You can identify the drugs, you can identify the lead molecules. So okay, in the literature survey, then you can take a preclinical studies, and once preclinical studies are effective, once preclinical studies are promising, then you can start our pre-formulation studies. Okay, simultaneously you have to do pre-formulation studies as well as that clinical trials. Okay, so you can easily get this information by literature survey or by literatures. So preliminary data, for example stability data, key solubility data, data solubility data, stability data, etc. Then you can optimize the molecule. Once you have the molecule in your hand, you can optimize the molecule, so depending upon the required physicochemical properties of drug. So you can develop that molecule in a different salt form, salt of organic acid, inorganic acid. You can make a solvate, citrate, etc, etc. You can make a prodrugs also. Then again, if you develop a prodrugs or solvate hydrates, you have to evaluate the drug. Okay, then formulation request. Okay, formulation, then you have to subject that drug to a formulation. Now in the formulation you have to study pre-formulation, pre-formulation parameters. So like bulk characterization, then crystallinity and polymorphism, then solubility analysis, then stability analysis, pK determination, etc, etc, drug expert interaction, etc, etc. Bulk, okay, first one is a physical characterization in the bulk properties of drug, solubility profile, stability profile of drug. Then formulation development in that compatibility of drug with the excipient, its stability studies, etc, etc. Then bioavailability, dissolution studies. If you are done with this study, then you can file NDA. Okay, so you can file IND, investigational new drug application. So phase first, that is known as the phase first formulation, IND stability, bioavailability and scale up. Then process research में आता है आपको improvement of yield, alternate route, producible. For example, if you are synthesized one drug, लेकिन वो synthesis के लिए आपको बहुत ही खर्च आ रहा है कि रॉ मटेरियल ये है और ये प्रोसेस है, प्रोसेस के बाद में आपको एक प्रोडक्ट मिलेगा, वो प्रोडक्ट सिंथेसाइज करने के लिए आपका जो खर्चा आ रहा है और वो सिंथेसाइज होने के बाद में उसका यील्ड कम है, यू कैन इंक्रीज द यील्ड, you can use alternate process, alternate route, okay, to produce that drug in a bulk in a economical way. Okay, so that is a process research. In process development, bulk scale up. Okay, process development, bulk scale up. Then after that, research, analytical research, development of analytical method like assay, then dissolution, etc. In analytical development, bulk clearance, toxicological potency, formulation assay, INDs, formulation stability. You can also do bioavailability and bioavailability studies like in vivo and in vitro. Toxicological studies. So it is nothing but the process through which IND is going away. So investigational new drug application, before applying for IND, you should have done with these studies. Before application of IND, you should have these studies. Okay? You should have studied this. Right. Now, once a drug has a promising effect in the animals, it assures once drug has a promising effect in the animal, that warranty, that assures the evaluation of the drug in humans. अगर आपके पास में अगर आपने कोई रिसर्च किया, फॉर एग्जांपल एक प्लांट है, उसमें आपने देखा कि अल्कोलाइड्स ग्लाइकोसाइड प्रीलिमिनरी टेस्ट से ये अल्कोलाइड्स का क्या इफेक्ट होता है एनिमल के ऊपर, नया कुछ अल्कोलाइड है, ये एंटी हाइपरटेंसिव है, आपने रिसर्च किया, ओके, if you got a promising effect in the animals, then only you can do its evaluation in the humans. So pre-formulation method deals with the physicochemical properties of a drug that could affect the drug's performance, then formulation, development, development in the particular dosage form. Right? So pre-formulation is nothing but the it deals with the different physicochemical properties. So what is a pre-formulation? Pre-formulation is nothing but the it deals with the physicochemical properties of drug that could affect the its formulation, performance, its therapeutic value, probable dosage form. So understanding the property, this property, जो formulation parameters है, understanding the formulation property helps in the formulation design and support the molecular modification if any required. अगर आपको मालूम है कि ये जो कंपाउंड है वो सोल्यूबल नहीं है वाटर में, वो लेस सोल्यूबल है, तो ऑब्वियसली यू कैन modify the method, you can make a solvents, you can make a hydrates, you can improve the solubility by enhancing, you can improve the solubility by co-solvent, etc., etc. So pre-formulation study helps to formulate or to design or to support any molecular changes or modification if any required. That's why you have to do pre-formulation study. तो ऐसा नहीं कि कोई ड्रग ले लिया इमीडिएटली उसका टैबलेट बना दिया, ऐसा नहीं होता. So you have to study lot of things. So flow properties, bulk characterization, polymorphism in that molecule, solubility analysis, stability, dissociation constant, etc., etc. You have to study a lot of things before the formulation. Okay, now these things, if you have studied, that assures this particular compound fit in this particular formulation. Okay? That's why pre-formulation is a confirmation that there is no significant hurdles for a compound development, formulation development. Now, why pre-formulation studies one has to do? Pre-formulation studies is done for to know the physico-chemical properties of a new drug. To know the physico-chemical properties of new drug. To know the physico-chemical properties of new drug. To know the kinetics and stability. Kinetics means absorption pattern, etc. Stability. To know the physicochemical properties, to know the kinetics and stability. Also, it helps to ensure the compatibility of the drug with a commonly used excipient. अगर वो आपका ड्रग जो है आपने जो सिंथेसाइज किया, जो आइसोलेट किया या फिर जो मॉडिफाई किया लीड मॉलिक्यूल से, वो ड्रग अगर आपका एक्सपिएंट के साथ में कंपेटेबल ही नहीं है, तो आप फॉर्मुलेट कैसे करोगे? सो प्री फॉर्मुलेशन स्टडी इज डन टू नो फिजिकल केमिकल प्रॉपर्टीज ऑफ ड्रग, टू नो द kinetics and stability, to ensure that your drug is compatible with the commonly used excipient. I hope I am audible to you. Am I audible? So pre-formulation chapter is again very important. As I told you, lot of things they used to ask from pre-formulation itself. So pre-formulation has a definitely impact on manufacturing, storage, then performance of drug. Okay, so pre-formulation is involved in the manufacturing. Okay, if your granules are not flowing from hopper to the tablet dye cavity, then what is the use of that granules? It is useless. So obviously, while designing a tablet dosage form, you have to study the particle size distribution, density, porosity, okay, of that powder. You have to analyze that powder flow properties, Hausner's ratio, Carr's index. You have to do angle of propose. So if it is not free flowing, then it will not flow from hopper to dye cavity for punching. So you will not get a proper tablet. You cannot ensure the uniformity of dosage. Okay? That's why pre-formulation has an impact on manufacturing, storage, performance of drug product. Pre-formulation provides the way by which a product should be processed. Pre-formulation क्या करता है अपने को? Pre-formulation study gives a particular way, gives a particular direction by which a drug product can be developed, or it can be processed, it can be stored, or it can be analyzed for quality. Now what involves in the pre-formulation, क्या क्या है उसमें? Primary findings and molecular optimization. First is primary finding and molecular optimization. So finding of known properties of dosage form. Okay, finding of known properties, dosage form, therapeutic area potential relative to comparative product. जो ऑलरेडी अवेलेबल है मार्केट में उसके कंपेरिजन में इसका थेरेपेटिक relative potential कितना है. In chemistry department should find out the molecular weakness and optimize the compound. Okay, तो केमिस्ट्री डिपार्टमेंट का काम क्या होता है कि मॉलिक्यूल में वीकनेस कहाँ पे है ना, मॉलिक्यूल को ऑप्टिमाइज कैसे करेंगे, मॉलिक्यूल का स्टेबिलिटी अगर नहीं है तो उसको एनहांसमेंट कैसे करेंगे, अगर वो इंस्टेबल है, अगर वो मेटाबोलाइज होता है तो उसका डीके मैकेनिज्म क्या है, डिग्रेडेशन कंडीशन कौन से, कितने टेम्परेचर का डिग्रेड होता है, कितना प्रेशर के ऊपर डिग्रेड होता है. It is a function of chemistry department. Chemistry department should find out the molecular weakness, optimize the molecule, probable decay mechanism, condition for degradation, etc. Now it is useful in the stability. Okay, it is useful for the stability test. Then method of stabilization of compound. Example, both there are different methods to stabilize compound. For example, अगर कोई एसिड है तो वो हाइड्रोलाइसिस होता है उसका एसिड का, एक काम करो उसको ईस्टर फॉर्म में बनाओ तो कम हाइड्रोलाइसिस होगा. So ester hydrolysis of aspirin, etc., etc. You can modify the molecule. Okay. Now this is just a background about the pre-formulation. Now solid state physical properties. Solid state physical properties. You have to analyze the compaction of powder. Compaction of powder. So in the solid state physical stability, physical property, solid state physical property, उसमें क्या क्या आएगा? Compaction properties कि कॉम्पैक्शन मतलब एक कॉम्पैक्टिबल है, मतलब कॉम्पैक्ट होता है नहीं होता है, कॉम्प्रेस होता है नहीं होता है. सो इन दैट यू आर गोइंग टू हैव मोलार वॉल्यूम, देन डेंसिटी, रिफ्रैक्टिव इंडेक्स, thermal and electrical conductivity, hygroscopicity. So in the compaction properties you have to study these properties of molecule. Okay, volume, then molar density, molar volume, density, refractive index, thermal and electrical conductivity, hygroscopicity. These are the compaction properties. Then thermodynamic properties. So in the thermodynamic properties, it means there is involvement of some kind of heat. In that melting point is there. Soluble, okay, sublimation is there. Melting point, sublimation, enthalpy, entropy, heat capacity, okay, free energy, then chemical potential, thermodynamic activity, vapor pressure, solubility. So in the thermodynamic property, you have to study these properties of chemicals. Thermodynamic में क्या पढ़ना है आपको? Melting point पढ़ना है, boiling point पढ़ना है, sublimation पढ़ना है, melting, boiling temperature, internal energy, that is enthalpy, heat capacity, entropy, free energy, chemical potential, then thermodynamic activity, vapor pressure, solubility, etc. So these are the thermodynamic properties. तो compaction properties में क्या है? मोलर वॉल्यूम, डेंसिटी, रिफ्रैक्टिव इंडेक्स, थर्मल एंड इलेक्ट्रिकल कंडक्टिविटी, हाइग्रोस्कोपिसिटी। सोलिबिलिटी किसमें आता है? थर्मोडायनेमिक में। अंडरस्टैंड, यू हैव टू अंडरस्टैंड। So free energy, solubility, vapor pressure, and this melting, sublimation, enthalpy, that comes under the thermodynamic properties of a solid state properties. So in that, next is the spectroscopic or analytical properties. So in that, UV, it is electronic properties, then vibrational properties by using IR, electronic properties by using UV, vibrational properties by using using IR, rotational by using microwave, nuclear spin, that is by NMR. Property of molecule, spectroscopic और एनालिटिकल प्रॉपर्टी, तो उसमें इलेक्ट्रॉनिक प्रॉपर्टी मतलब इलेक्ट्रॉनिक एक्साइटेशन is studied by UV spectroscopy. Molecular vibration are studied by IR spectroscopy. Rotational spectroscopy is nothing but the microwave. There is a spin change in the spin state in the NMR, okay, nuclear spin, the NMR. So these are the spectroscopic analytical property. So compact, first one is compaction property, second one is thermodynamic property, third one is the spectroscopic analytical properties. Now fourth one is the kinetic properties. Kinetic properties. So in that you are having dissolution rate, solid state reaction rate, and stability. ये kinetic properties होगा solid. Then surface properties में क्या आएगा फिर? Surface force, surface, okay, and surface force, surface and interfacial tension, crystallinity, है ना? ये क्या है? Surface property है. The next mechanical properties. So in that hardness, tensile strength of material, compatibility है. Then flow properties of material and blending of that material. Okay. Now salt formation. Salt formation means what? So when a compound is there, if any organic acid is there, so organic compound by abstracting the proton from ionized drug and neutralizing it with the counter ion will form a salt. Say, say salicylic acid, sodium hydroxide, sodium salicylic salt बनता है. Generally, organic acid are more soluble in the organic salts are soluble in the water. जो free acid और free base जो होता है, that is not soluble in the water. So salt is soluble in the water. So generally, salts are more soluble in the water than unionized drug. So ionized drug is more soluble in the water. Unionized drug is soluble in the lipid, which offers the higher. So salt form of drug will offers the higher dissolution and bioavailability. तो salt form जो है ड्रग का वो क्या है? सोल्यूबल होता है वाटर में. दैट्स व्हाई आपने देखा होगा मोस्ट ऑफ ड्रग लाइक कैफीन साइट्रेट है ना, इपेड्रिन हाइड्रोक्लोराइड, मॉर्फिन सल्फेट. व्हाई दीज कंपाउंड्स दे आर formed into a salt form or a salt formation? Because the free bases or acid of these compounds, they are not having solubility in the water. To enhance their solubility, ultimately to increase the bioavailability, these drugs are formulated in the salt form. Okay, so caffeine citrate, morphine hydrochloride, epatine hydrochloride, etc., salt formation. So common anion like hydrochloride, bromide, मतलब acid कौन से थे? Common acid like hydrochloric acid or hydrochloride, bromide, chloride, iodide, citrate, malleate, phosphate, sulfate, pampate, and cations like magnesium, calcium, sodium, zinc are used as cations for the salt formation. Because salt अगर है तो वो soluble रहेगा, free form soluble नहीं हो रहा है. That's why salt formation is important. Pre-formulation studies, they are done under main three headings. Pre-formulation studies are done under main three headings. First heading is nothing but the solubility studies. In that, pKa determination, dissociation constant determination, then pH solubility profile, then common ion effect, then effect of temperature on solubility, solubilization mechanisms, then partition coefficient, dissolution study of that drug. So these are the solubility studies. Okay? तो solubility studies में क्या है? pKa determination आएगा, pH solubility profile आएगा, common ion effect आएगा, effect of temperature on solubility, solubilization mechanism आएंगे, को सॉल्वेंसी आएगा फिनोमेना, पार्टीशन कोएफिशिएंट, डिसोल्यूशन, ये सब किसके अंदर आता है? सोलिबिलिटी स्टडीज के अंदर. दूसरा आता है आपका बल्क कैरेक्टराइजेशन. सेकंड में हेडिंग आता है बल्क कैरेक्टराइजेशन. इन दैट यू हैव टू स्टडी क्रिस्टलिटी एंड पॉलीमॉर्फिज्म ऑफ कंपाउंड. ओके, देन hygroscopicity, deliquescency, efflorescency, then micrometric properties like particle size characterization, density, porosity, flow properties of powder. And in the third heading आता है stability analysis. In that solution stability is there, solid state stability is there, drug excipient compatibility is there, and long-term stability, intermediate term stability, and short-term stability, accelerated stability studies are there. So you have to understand these are the main headings under this pre-population chapter. We have to study all these points. We have to study all these points. Believe me, इसके ऊपर क्वेश्चन आते ही आते हैं, ठीक है. जैसे बहुत बार क्वेश्चन आ गया को सॉल्वेंसी, मोस्ट कॉमनली यूज्ड को सॉल्वेंट है ना, पीके है ना, कॉमन आयनिफाइड से आता है, डिसोल्यूशन, पार्टीशन कोएफिशिएंट. तो क्वेश्चन आते हैं उसके ऊपर. we'll take one by one. First one is a bulk characterization. Bulk characterization, in that we'll study crystallinity and polymorphism. Crystallinity, crystallinity and polymorphism. We have already seen the crystallization process in the pharmaceutical engineering, and in that we have seen how there is a formation of crystal. How there is a formation of crystal. Polymorphism means what? Polymorphism is nothing but the property of compound to crystallize in a different crystal form. That is a polymorphism. If a same compound is having different crystal forms, for example, आपका आता है ना, chloramphenicol palmitate. Chloramphenicol palmitate will crystallize in a three different form. Chloramphenicol palmitate A, B, C, D. Now these polymorph, they are known as a polymorph. These polymorphic form of compound will have different physicochemical properties. तो उनका physicochemical properties different होता है, and that's why they will affect the formulation. So for example, जैसे आपका और एक example, so suppository में आता है आपका cocoa butter. So suppository basis they are known as a cocoa butter. So cocoa butter will crystallize in a different crystallization form. So known as a polymorphic form, alpha, beta, gamma, delta. Right? So out of that, beta form is a stable. So if you prepare a suppository by using cocoa butter base, and if that alpha form crystallizes, so what will happen? That alpha form will melt at a lower temperature, है ना. तो जैसे अल्फा फॉर्म होता है, आपने सपोजिटरी बनाया तो आपके हाथ में ही मेल्ट हो जाएगा वो, because alpha form will melt at a, it has a low melting point. So beta form is stable. Beta form will melt at a 37 degree. That is body temperature. So that's why polymorphism effect or polymorphism affect the formulation. Now what is polymorphism? Existence of compound in a different crystalline form known as a polymorphism, or if a compound crystallizes in a different crystal form known as a polymorphism. So what? So you have to take a compound, you have to observe under a microscope. अगर एक जो भी है आपके पास कंपाउंड, वो आपको माइक्रोस्कोप के अंडर ऑब्जर्व करना है. सो कंपाउंड अंडर माइक्रोस्कोप. नाउ इफ इट इज अ ट्रांसपेरेंट कंपाउंड, now if the compound is a transparent, if compound is a transparent, it is known as a bifringes. Okay, and again bifringes are of two type. Okay, bifringes means they are having refractive index. Transparent कब होता है compound? It has a refractive index, so that's why bifringes. Now if the compound has a more than one refractive index, if compound has a more than one refractive index, it is known as anisotropic. One plus refractive index, more than one refractive index. if compound has more than one refractive index known as anisotropic, और वही compound और एक refractive index है, it is known as isotropic. If compound has a one refractive index, it is known as isotropic. Okay? Now anisotropy is further divided into two types. So anisotropy में भी आता है uniaxial and biaxial. Uniaxial and biaxial. Uniaxial means anisotropic compound having two refractive index known as uniaxial, and anisotropic compound having three refractive index known as a biaxial. Right? So if you observe a compound under a microscope, it may be either transparent or it may be either non-transparent. अगर कंपाउंड को आपने माइक्रोस्कोप के अंदर ऑब्जर्व किया तो एक तो उससे लाइट पास होगा या फिर नहीं होगा. अगर लाइट पास आउट होता है मतलब इट इज अ ट्रांसपेरेंट, इट मींस इट इज अ बायफ्रेंजेस. उसके उसमें क्या है, रिफ्रैक्टिव इंडेक्स है. अगर उसमें एक से ज्यादा रिफ्रैक्टिव इंडेक्स है उसको बोलेंगे अपन एनिसोट्रोपिक. अगर एक ही रिफ्रैक्टिव इंडेक्स है तो उसको बोलेंगे अपन आइसोट्रोपिक कंपाउंड. ठीक है. अगर अनिसोट्रोपी में दो रिफ्रैक्टिव इंडेक्स है उसको बोलेंगे अपन यूनिएक्सियल. अगर तीन है तो उसको बोलेंगे अपन बायएक्सियल. ओके. नाउ इफ कंपाउंड इज अ नॉन ट्रांसपेरेंट, अगर कंपाउंड क्या है, नॉन ट्रांसपेरेंट है, ट्रांसपेरेंट नहीं है, राइट. नाउ इट कैन बी डिस्क्राइब्ड बाय टू टर्म्स. इट कैन बी डिस्क्राइब्ड बाय two terms. What is the term? First term is nothing but the internal structure of that compound and external structure of that compound. External structure जो होता है उसको अपन बोलते हैं हैबिट. So external structure of compound is known as a habit. इसके ऊपर क्वेश्चन आ चुका है. External structure of compound is nothing but the habit. Okay, habit. External structure of compound is nothing but the habit. Are you getting or not? समझ रहे आपको? Am I audible? So if compound is non-transparent, that means if compound is non-transparent, so it has an external structure known as a habit, and internal structure is known as a internal structure is either further divided into two type. जो कंपाउंड अगर है ट्रांसपेरेंट है तो बाय फ्रिंजेस होता है, उसमें अनिसोटॉपी अनिसोटॉपी होता है. अगर ट्रांसपेरेंट नहीं है तो एक तो एक्सटर्नल स्ट्रक्चर होगा इसका, उसको अपन बोलते हैं हैबिट. एक्सटर्नल स्ट्रक्चर ऑफ कंपाउंड इज नोन एज हैबिट. इसके ऊपर क्वेश्चन आ चुका है. एंड इंटरनल स्ट्रक्चर इज डिवाइडेड इनटू टू टाइप. इट मे बी आइदर क्रिस्टलाइन और amorphous. It may be either crystalline or amorphous. Now crystalline or amorphous. Now, crystal may be either crystal, it may have crystalline structure or amorphous structure. So internal structure of compound. Okay. Now crystalline means what? So in the crystal, during the formation of crystal, there is a systematic arrangement of particle in the crystal lattice. In the crystalline form. So crystalline form में क्या है? There is a systemic arrangement of particles in the crystal lattice, and that's why you can say this system is stable. It has a low energy and low solubility. So in the crystalline there is a systematic arrangement of particle in the crystal lattice. That's why it has a low energy, system is stable, and it has a low solubility. Now in case of amorphous means what? So there is a random arrangement of particles in the crystal lattice, and that system is unstable. It has a high energy, therefore it has a high solubility. तो क्वेश्चन आता है which of the following form has a highest solubility? ऑप्शन में आएगा आपको ऑप्शन ए. which form of compound has a highest solubility? ऑप्शन ए ना. ऑप्शन ए में आएगा आपको क्रिस्टल फॉर्म. ऑप्शन बी में आएगा amorphous फॉर्म. ऑप्शन सी में आएगा मेटास्टेबल फॉर्म. ऑप्शन डी में आएगा ऑल ऑफ द अबव. है ना. एक सिंपल क्वेश्चन अगर पूछेंगे तो ऐसे पूछेंगे. So in that case, amorphous form of compound has a highest solubility, because in the amorphous there is a random arrangement of particle, therefore the system is unstable. It has a higher energy, and that's why it has a highest solubility. Remember. Now crystalline में क्या होता है? crystalline form जो होता है वो stable होता है. उसमें systematic arrangement of particles in the crystal lattice होता है, और वो उसमें energy कम होता है. System stable होता है. That's why it has a low solubility. बहुत सारे लोगों को लगता है क्रिस्टलाइन फॉर्म जल्दी सोल्यूबल होगा. नहीं, amorphous form is a having more solubility. That's why certain drugs they are converted from crystalline to amorphous to enhance their solubility. So one amorphous formation is one of the method to enhance the solubility. Now now how you can prepare amorphous form of drug? Amorphous form of drug can be prepared by either rapid precipitation, then lyophilization, and rapid cooling of liquid during crystallization. So amorphous form can be prepared by this. इसके बीच में एक metastable form होता है. metastable form. So in between crystalline amorphous there is one metastable form. crystalline amorphous metastable form. इसके बीच में होता है metastable. crystalline form, amorphous form, and metastable. Now amorphous form has a highest solubility. Remember, amorphous form of compound will have a highest solubility. Crystalline form will have a lowest solubility, and in between that you have metastable form. तो आपको और एक तरीके का क्वेश्चन आता है, arrange the following form of compound in their increasing order of solubility. है ना. तो आपको देंगे crystalline is more than amorphous is more than metastable. दूसरे ऑप्शन में देंगे क्रिस्टलाइन इज मोर देन मेटास्टेबल इज लेस देन amorphous. और ऐसे कुछ घुमा के फिर के ऑप्शन लेंगे. सो रिमेंबर, amorphous form of compound has a highest solubility. सबसे ज्यादा soluble ये होता है वाटर में. उसके बाद में मेटास्टेबल होता है, उसके बाद में क्रिस्टलाइन. रिमेंबर दिस इज solubility, है ना, solubility of different doses. Crystalline compound are other. Crystalline compound is further divided into two types: single polymer and molecular adduct. And single polymer further divided into two types: monotropic and anisotropic. Monotropic means all polymorphs are unstable at a temperature and pressure. Anisotropic means all polymorphs are isolable. Okay. Now in the molecular adduct मतलब क्या, molecular adduct मतलब कुछ तो ऐड हुआ है, क्या ऐड हुआ है, मे बी सॉल्वेंट ऐड हुआ है या फिर वाटर ऐड हुआ है, जो भी सॉल्वेंट ऑफ क्रिस्टलाइजेशन है. सो सॉल्वेंट ऑफ क्रिस्टलाइजेशन मोस्ट ऑफ टाइम इट इज वाटर, molecular adduct. Now if it is a stoichiometric and non stoichiometric, तो जो ऐड हो चुका है ड्यूरिंग क्रिस्टलाइजेशन, तो सॉल्वेंट ड्यूरिंग क्रिस्टलाइजेशन, solvent of, okay, whatever solvent is there in the crystallization, that solvent may get entrapped between the crystal, known as a molecular adduct. During the crystallization process, the solvent may get entrapped into the crystal, known as a molecular adduct. Now, if that solvent is water, then it is a, okay, if that solvent water भी हो सकता है, दूसरा भी हो सकता है. So, solvent of crystallization, if it gets added to a molecule in a stoichiometric form. Stoichiometric form means 1:1. Stoichiometric form मतलब क्या, 1:1. The solvent per crystallization, it gets entrapped into a molecule, into a crystal, 1:1 proportion, known as a stoichiometric, and that is known as a solvents. Stoichiometric अगर हो तो उसको बोलेंगे solvents. Solvents मतलब क्या, जो solvent है वो ट्रैप हो चुका है, solvent is added in the crystal in 1:1 proportion, known as solvents. Now if that solvent is nothing but the water, अगर वो solvent water है तो उसको बोलेंगे hydrate. If solvent is water, then it is hydrate. Solvents and hydrate are the stoichiometric molecular adduct for the crystalline state. Have to remember. Now non-stoichiometric means not in a proportion, not in a 1:1 proportion. Proportion 1:1 नहीं होगा. So non-stoichiometric in that, so depending on the shape, there may be a development of cage-like structure. There might be a development of layer-like structure. There might be a development of channel-like structure. GPAT 2023 में इसके ऊपर क्वेश्चन है. Channels. Molecular adduct having channel-like shape. They are known as non-stoichiometric channels. Cage, layer, channel, ये तीनों ऑप्शन था. मतलब क्वेश्चन इसके ऊपर भी आ चुका है. आर यू गेटिंग और नॉट, समझ रहे आपको. एग्जांपल्स ऑफ क्रिस्टल हैबिट मींस व्हाट, हैबिट मींस external structure of your crystal, external structure of crystal known as a habit. It may be depending on the shape, it may be acicular or acicular. इसका डिस्क्रिप्शन क्या है इसका, they are the elongated prism or needle like structure. एसिकुलर क्रिस्टल हैबिट मतलब क्या, elongated होता है, प्रिज्म और needle like होता है. Then angular habit, sharp edge, उसका ब्लेड जैसे sharp edge होगा, roughly polyhedral in a shape. Then bladed मतलब क्या, flattened acicular, crystalline, fully developed geometric shape in the, in the fluid. Okay. Then dendritic shape भी होता है, dendritic, branch twist line, fibrous भी हो सकता है. जैसे आपने अगर गन्ने को तोड़ दिया बीच में से, गन्ने में कैसे फाइबर निकलते वैसे. So fibrous, regular or irregular thread like structure. Then flaky or platy, plate or salt like structure. Granular structure, so equidimensional irregular shape. Irregular crystal habit हो सकता है, like as asymmetry. Nodular हो सकता है, rounded irregular shape. Prismatic हो सकता है, geometric prism. Spherical हो सकता है, globule shape. Okay. Now, polymorphism means what? Ability of compound to crystallize in a one or more than one crystalline form. Ability of compound to crystallize in a different crystal form. It is a polymorphism. It affects the solubility and stability of those form. In turn, affect the bioavailability. Because जैसे अपने देखा है कि crystalline form is a less soluble in water, amorphous form is a more soluble. तो amorphous form अगर है कंपाउंड का, ड्रग का आपके डोसेज फॉर्म में, तो उसका बायो अवेलेबिलिटी ज़्यादा होगा क्योंकि उसको सोलिबिलिटी ज़्यादा है. अगर क्रिस्टलाइन फॉर्म है तो वो सोलिबिलिटी उसका कम है, तो बायो अवेलेबिलिटी उसका कम रहेगा. तो इट अफेक्ट्स द स्टेबिलिटी एज वेल एज सोलिबिलिटी ऑफ डोसेज फॉर्म, इन टर्म्स बायो अवेलेबिलिटी. फॉर एग्जांपल, chloromphenical palmitate has a 3 crystalline form, A, B and C. So amorphous form of which, okay, okay, among these, amongst this, amongst this form, beta form is more stable, P form is more stable of clone panicle permitted. Now, physicochemical properties of drug, they vary with the internal structure of drug of a solid drug. Example, melting point वो change हो सकता है, internal structure change हो पाए तो. Physicochemical properties of a drug varies with the internal structure of drug. Example, melting point will vary, then hardness, solubility, optical properties, okay, vapor pressure. This, so these properties, vapor pressure, optical properties, solubility, they can vary with the internal structure of drug. That's why we have to study polymorphism. Now you can see over here, it's a platy external laboratory is our platy-like structure is this. Equant or mossy, needle or acicular crystallite, okay, tubular crystallite, prismatic crystallite, bladed crystallite. Now methods, different methods for quantitative analysis of physical properties of drug. तो कैसे physical properties of analyze कर सकते हो, different method. So microscopy, by using microscopy you can observe the component under microscope, you can determine the polymorphism, you can measure, you can also determine the crystal habit, etc. So 1 milligram sample is enough to study physical properties of drug by microscopy. Okay, second होता है आपका फ्यूजन मेथड और हॉट स्टेज माइक्रोस्कोपी. इसमें हॉट स्टेज माइक्रोस्कोपी किसके लिए आएगा? मेल्टिंग प्रॉपर्टीज ऑफ ड्रग, melting property, enthalpy. 1 milligram sample is enough. Differential scanning calorimetry, differential thermal analysis. So 2 to 5 milligram of sample is required for this differential scanning calorimetry and differential thermal analysis (DTA). So using this you can determine DCC और DSA क्या मिलता है आपको, melting point कर सकते हो आप, है ना, melting point, ठीक है, then boiling point, उसके बाद में heat of formation, है ना, heat of formation, ये सब चेक कैसे कर सकते हो, DSC differential scanning calorimetry and differential thermal analysis. Heat of fusion, etc. By using IR spectroscopy, you can measure the water content of that drug. So 2 to 20 milligram sample is required for IR. By using X-ray diffraction, you can study the crystal structure. So 500 milligram of sample is required for X-ray diffraction. Crystal structure and crystal structure, plane of crystal. Scanning electron microscope (ACM). 2 milligram of sample is required. थर्मोग्राविमेट्रिक एनालिसिस के लिए आपको 10 मिलीग्राम सैंपल लगेगा. सॉल्युबिलिटी डिसोल्यूशन एनालिसिस के लिए आपको few milligram to gram sample लगता है. So this is different techniques which are used in the study of physicochemical properties of drug. Physicochemical properties of, again you can see over here same thing. Methods and applications, qualitative analysis of physical and chemical properties of drug. So methods, for example, पहले है आपका microscopy. So it is used to study the structure of substance, crystal of substance morphology, as in a chart we have seen already. Then fusion method or hot stage microscopy is used to study the crystal melting properties and all that. Then DSC, DTA, differential scanning calorimetry, differential thermal analysis. So they are used to study the polymorphism. They are used to study the polymorphism, crystal purity. इसके ऊपर एक क्वेश्चन आ चुका है, crystal purity, sharp, okay, sharp peak in the DSC, है ना. DSC में अगर कोई ऐसा एकदम ऐसा sharp peak आता है, ये हो गया broad peak, ये broad peak होगा, ऐसा कुछ sharp peak आता है. Sharp peak means more will be the sharper the peak, more will be the pure of crystal. Okay? So crystal purity, sharp peak indicate the pure crystal in DSC. नाइपर में इसके ऊपर क्वेश्चन आ चुके हैं. नाइपर में ब्रॉड पीक होता है, पीक्स आर ऑफ टू टाइप, ब्रॉड पीक एंड शार्प पीक. अगर शार्प आता है पीक तो उसका मतलब क्रिस्टल क्या है, प्योर है. से ब्रॉड पीक कैसा होगा, ये ऐसा होगा ब्रॉड पीक. शार्प पीक कैसा होगा, मतलब sharp peak. IR spectroscopy, it is used to measure the water content of crystal. Okay. X-ray diffraction to study the crystal shape and outer surface of crystal. Scanning electron microscopy to study the crystal morphology. Thermogravity analysis for the study of crystal. NMR, study of crystal. HPLC, HPLC is used which is the stability indicating assay used for the stability studies. और इसके ऊपर भी नाइप में क्वेश्चन आ चुका है. स्टेबिलिटी इंडिकेटिंग assay is nothing but the HPLC. Solubility and dissolution के लिए बायो अवेलेबिलिटी है ना. Solution dissolution studies or dissolution solubility studies, you can check the bio availability. Now moisture content in the compound. Moisture content कैसे measure करते हो आप? Moisture content you can measure moisture content in a different technique. Moisture content you can measure by different technique. जैसे होता है Curl Fisher titration. So Curl Fisher reagent. First one is known as Curl Fisher reagent. You can measure the moisture content by using Curl Fisher titration. You can measure moisture by using toluene distillation method. You can measure moisture by using IR spectroscopy. There are different methods for measurement of moisture content. So based upon that, compound is divided into three types. First one is known as a hygroscopic compound. तो पहला होता है hygroscopic compound. So what do you mean by hygroscopic compound? So a compound or substance that will absorb moisture from air. Okay, when it is exposed to a humid condition. When compound is exposed to humid condition, it will absorb the moisture from air. It will absorb the moisture from air. If you expose that compound to higher humidity values, so it will absorb the compound will absorb the moisture from environment, and there is an increase in the weight of that compound. Okay. Now next term होता है deliquescent. What do you mean by deliquescent? Hygroscopy means a substance that will absorb the moisture from environment, and there is an increase in the weight. And deliquescent means this compound will absorb a sufficient moisture to get dissolved or liquefied. तो पहला वाला जो केस है हाइग्रोस्कोपिक, तो ये कंपाउंड भी क्या है, वाटर एब्सॉर्ब करता है मॉइस्चर में से. अगर आप उसको हायर ह्यूमिड कंडीशन पे, if you expose that compound to a humid condition, it will absorb the moisture from air, and there is increase in the weight of that compound. That is hygroscopic. Now deliquescent mean, so these are also compound which absorb the moisture from air, but to a such an extent that they will get liquefied. पूरा लिक्विड हो जाए. जैसे बारिश के दिनों में अगर आपने नमक है ना, साल्ट अगर ओपन रख दिया तो उसका सब क्या होता है, पानी हो जाता है. दैट इज अ डेलिक्वेशन. नाउ अनदर टर्म इज देयर नोन एज इफ्लोरोसेंट मेटल. इफ्लोरोसेंट मतलब क्या? इफ्लोरोसेंट क्या होता है? इफ्लोरोसेंट मींस ए मटेरियल व्हिच विल रिलीज द वाटर. इट विल रिलीज द वाटर व्हेन इट इज एक्सपोज्ड टू ह्यूमिड कंडीशन. ये क्या करेगा? वाटर ऑफ क्रिस्टलाइजेशन को छोड़ देगा. इट विल रिलीज द वाटर ऑफ क्रिस्टलाइजेशन व्हेन एक्सपोज्ड टू ह्यूमिड कंडीशन. इसके ऊपर जीपैट 2023 में क्वेश्चन आ चुका है. ये आई थिंक मैंने पहले भी बोला होगा आपको, ड्राइंग चैप्टर में बोला होगा. सो डेलिक्वेसेंट, हाइग्रोस्कोपिक एंड इफ्लोरोसेंट. इफ्लोरोसेंट मेटल होता है कैफीन. इसके बाद में आपका सोडियम वेलपोरेट. So changes in the moisture level that significantly affect the chemical stability, flowability, compactability of the compound. अगर कंपाउंड में मॉइस्चर ज़्यादा है तो ऑब्वियसली उसका केमिकल स्टेबिलिटी क्या होगा, वो जल्दी ही हाइड्रोलिस होगा सकता है. इफ मॉइस्चर इज प्रेजेंट इन द कंपाउंड तो इट मे लीड्स टू हाइड्रोलिसिस. अगर कंपाउंड में मॉइस्चर है तो हाइड्रोलिसिस हो सकता है आपका. तो केमिकल स्टेबिलिटी क्या होगा, उसका अफेक्ट होगा. देन इफ देयर इज अ मॉइस्चर इन द कंपाउंड सो इट विल नॉट फ्री फ्लो, तो फ्लोएबिलिटी अफेक्ट होगा. इफ देयर इज अ मॉइस्चर इन द कंपाउंड सो दैट कंपाउंड अफेक्ट द कॉम्पैक्टेबिलिटी. उसका कॉम्पैक्शन पैरामीटर जो है ना वो अफेक्ट होगा, क्योंकि वो जल्दी कॉम्प्रेस हो जाएगा और वो बहुत ही हार्ड केक बनेगा. So changes in the moisture level significantly affect the stability, flowability and compatibility. Now European Pharmacopoeial Technical Guide has classified degree of hygroscopicity into the four classes based upon the static method. So you have to store the compound. At 25 degrees Celsius for 24 hours at a 80% relative humidity. ओके. तो हाइग्रोस्कोपिक कंपाउंड मतलब क्या, वो मॉइस्चर अब्सॉर्ब करेगा, है ना. तो आपको क्या करना है कि यूरोपियन टेक्निकल गाइड जो है उनके अकॉर्डिंगली यूरोपियन फार्माकोपिया टेक्निकल गाइड दे आर क्लासिफाइड हाइग्रोस्कोपी इन 4 पार्ट बेस्ड अपॉन द स्टैटिक मेथड. क्या है, आपको एक प्री वेड है, जैसे मान लीजिए 1 ग्राम कंपाउंड लेना है. यू हैव टू एक्सपोज दैट कंपाउंड at 25 degrees Celsius फॉर 24 आवर्स एट 80% ह्यूमिडिटी। तो उसको स्टेबिलिटी चेंबर में रखना है। यू हैव टू सेट 25 डिग्री सेल्सियस टेम्परेचर, है ना, and 80% relative humidity, and then you have to, okay, then you have to after 24 hours, you have to take that compound and you have to measure the weight। पहले तो वेट किया आपने, जैसे मान लीजिए 1 ग्राम वेट लिया था, बाद में 24 आवर्स के बाद में भी इसका वेट लेना है ना। इसका वेट क्या होगा, ऑब्वियसली हाइड्रोस्कोपिक कंपाउंड है, उसका वेट बढ़ेगा, है ना, कुछ अमाउंट में, है ना, उसको हाइग्रोस्कोपी बोलेंगे। अगर वेरी हाइग्रोस्कोपी है तो उसका वेट थोड़ा ज़्यादा बढ़ेगा, और अगर डेलिक्वेशन है तो उसका वेट और भी मतलब लिक्विड बन जाएगा। सो बेस्ड अपॉन दैट, सो यूरोपियन फार्माकोपिकल टेक्निकल गाइड दे हैज क्लासिफाइड हाइग्रोस्कोपिसिटी इन 4 classes based upon the static method। So you have to pre-weigh the compound, you have to subject the compound at a control condition like 25 degrees Celsius temperature, 24 hours time, 80% relative humidity। Now you have to check the weight of that compound। If increase in the weight of that compound, अगर वो compound का weight increase हो गया, if increase in the weight of compound is more than 0.2% weight by weight but less than 2%, less than 2%, so after subjecting a compound to a control condition, that is 25 degrees Celsius for 24 hours, 80% relative humidity, now you have to weigh the compound। If increase in the weight of that compound is more than 0.2 weight by weight but less than 2%, then it is known as a slightly hygroscopic, it is known as a slightly hygroscopic। तो कंपाउंड को स्लाइटली hygroscopic कब बोलेंगे, अगर उसका वेट इंक्रीज होगा कितना, लेस देन 2%, लेस देन 2% weight of weight। if increase in the weight is less than 2%, it is known as a slightly hygroscopic। if increase in the weight is more than 0.2% but less than 15%, it is known as a hygroscopic। इसके ऊपर क्वेश्चन आ चुका है GPAT 2021 में। now if increase in the weight is more than 15% weight by weight, it is known as a very hygroscopic, very hygroscopic। deliquescence will absorb sufficient moisture to become a solution or liquid, deliquescence। so इसके ऊपर ये याद ही रखना है आपको। कैसे याद रखना है, सिंपल है, देखो, है ना, 25 डिग्री सेल्सियस 80% रिलेटिव ह्यूमिडिटी 24 आवर्स में कंपाउंड को एक्सपोज करना है और वेट मेजर करना है। इफ वेट इज ओरिजिनल वेट है 1 ग्राम, if increase in the weight is less than 2%, so it is a slightly hygroscopic। if increase in the weight is more than 2% but less than 15%, it is a hygroscopic। if increase in the weight is more than 15% weight by weight, it is a very hygroscopic। एंड अगर वो लिक्विड बनता है तो डेरिवेशन, सिंपल है, क्वेश्चन आ चुका है इसको। फॉलोइंग मेथड्स आर यूज्ड टू डिटरमाइन द मॉइस्चर कंटेंट। मॉइस्चर कंटेंट के लिए कितने मेथड है, कितने मेथड है। सो थर्मोग्रामेट्रिक एनालिसिस टीजीए, थर्मोग्रामेट्रिक एनालिसिस, देन गैस क्रोमेटोग्राफी, गैस क्रोमेटोग्राफी से भी आप मॉइस्चर कंटेंट डिटरमाइन कर सकते हो। देन ग्रेविमेट्री, ओके, देन कर्ल फिशर टाइट्रेशन, केएफ रिएजन, उसमें रीजन में क्या क्या होता है इसके, ये भी याद रखना है, रीजन में क्या होता है इसके। सो इन द कर्ल फिशर रिएजन यू हैव pyridine, pyridine that is amine, pyridine, then you have methanol, pyridine, methanol, then you have iodine and potassium iodide। pyridine, methanol, iodide, potassium iodide, they are the reason of our कर्ल फिशर, याद रखना है। नाउ वेंट ऑफ फ्लॉट, इसके ऊपर भी क्वेश्चन आ चुके हैं बहुत बार। व्हाट इज वेंट ऑफ फ्लॉट, वेंट ऑफ फ्लॉट क्या होता है। इफ यू प्लॉट ए temperature versus molar solubility of compound, if you plot temperature versus molar solubility of compound, then this plot is known as a Ventop plot। molar solubility क्या होता है, what is molar solubility, temperature at which solubility and vapor pressure of polymorphs are same, the temperature at which solubility and vapor pressure of all polymorphs, अगर एक कंपाउंड है उसके पॉलीमॉर्फ 2-3 पॉलीमॉर्फ है, पॉलीमॉर्फ 1, पॉलीमॉर्फ 2, पॉलीमॉर्फ 3, so temperature at which solubility and vapor pressure of polymorph are same, then that is known as a molar solubility। And graph of temperature versus molar solubility is nothing but the Ventop plot। Ventop plot, इसके ऊपर क्वेश्चन आ चुका है। Now what is bridging solvent? So it is a solvent used to convert one polymorph to another polymorph। जैसे एक polymorph में आपके पास में मान लीजिए अल्फा polymorph है आपके पास, वो स्टेबल ने आपको बीटा करना है। The solvent which is used to convert one polymorph to another polymorph known as a known as a bridging solvent. Known as a bridging solvent. नाइपर के लिए क्वेश्चन आ चुका है ये. Then fine particle characterization. Fine particle characterization. इसमें क्या क्या है? Bulk flow, formulation, homogeneity, the surface area, control process like distribution, and a chemical reactivity which is affected by size, shape, morphology. Now, what are the different methods which are used for the particle characterization? Methods for particle characterization. So we have seen already in the micrometrics. So sieving method is the sieve analysis. Particle size more than 100 microns. Then we have light microscopy or known as optical microscopy. We have seen the culture counter method in the physical pharmacy. We have seen the HIAC, that is nothing but the stream counting method. Adsorption method is there, and in the adsorption method, uh, adsorption method is there. Then sedimentation method is there. In the in the sediment in the sedimentation method, you have Stokes law. Now after that you have adsorption method to measure the particle size and particle surface area. That is nothing but the adsorption method. In that you are having this equation, Bate equation, Brunner, Emmett and Taylor equation. So liquid nitrogen at a 196 degree, liquid nitrogen at a 196 degree is already to adsorb on a powder to form a monolayer, and one nitrogen atom is equal to 16 angstrom unit. There is one apparatus which is used to measure the surface area of particle by using adsorption method known as a quantasol. Known as a quantasol. ये अपने फिजिकल फार्मेसी में देखा है क्वांटासॉल. क्वेश्चन उसके ऊपर आ चुका है. Now scanning electron microscope surface morphology, surface texture, it can be studied by scanning electron microscope. Compound is gold plated to make a superconductive. स्कैनिंग इलेक्ट्रॉन माइक्रोस्कोप के नीचे जो भी कंपाउंड अंडर इन्वेस्टिगेशन है उसको गोल्ड प्लेटिंग करना है. क्यों? कंडक्टिविटी बढ़ता है उसका. बल्क डेंसिटी क्या होता है? बल्क डेंसिटी इज नथिंग बट द सिंपल मास अपॉन वॉल्यूम. It is important. If bulk density is very important because high dose of capsule is required. In case of high dose of capsule is, तो कैप्सूल में उतना आना चाहिए. बल्क डेंसिटी ज़्यादा है तो apparent bulk density ग्राम पर ml is calculated on a 40 mesh received powder. कैलकुलेटर कैसे करते हैं? पहले तो जो पाउडर है वो 40 mesh से पास करना है. जो नीचे पाउडर आएगा आपको उसके ही ऊपर उसका वॉल्यूम एंड मास मेजर करना है. और मास अपॉन वॉल्यूम क्या होगा? Density. True density, we have seen this. Bulk density minus void volume. Tap density is measured by, or it is calculated by pouring a known quantity of powder in the graduated cylinder and then tapping not less than 1000 times in the taper density. Now flow properties of powder is determined by two-three methods. First one is a compressibility index, known as a percent compressibility index. Percent compressibility index. Angle of repose. Then Hausner's ratio. Hausner's ratio. They will determine the flow properties of powder, right? Powder may be either free flowing powder हो सकता है या फिर नॉन फ्री फ्लोइंग को अपन बोलते हैं कोहेसिव powder. पाउडर में भी फ्री फ्लोइंग और नॉन फ्री फ्लोइंग मतलब कोहेसिव मतलब उसके इंटर पार्टिकुलर अट्रैक्शन ज्यादा होते हैं उसमें एंड ऐसा उसका फ्लो क्या होता है? कम होता है. फ्री फ्लोइंग में इंटर पार्टिकुलर अट्रैक्शन कम होता है. नाउ कॉम्प्रेसिबिलिटी इंडेक्स also known as a Carr's index. Compressibility compressibility index or also known as a Carr's index. So it is a measure of flow properties of powder. Percent compressibility और Carr's index और Carr's consolidation index इसको ही बोलते हैं Carr's consolidation index. Percent compressibility is equal to rho t minus rho 0 divided by rho t into 100. Rho क्या है? Rho t is the tap density and rho 0 is the bulk density. So compressibility is nothing but the tap density divided by bulk density. Tap density minus bulk density divided by tap density into 100. That is the Carr's index. Now Hausner's ratio, it is nothing but the tap density divided by pore density. Tap density divided by pore density or bulk density. Hausner's ratio again is the parameter for flow properties, and it will range from 1.2, which is for free flowing powder, and 1.6, which is for cohesive powder. Now you can see over here. Now Cars index of or percent compressibility, or simply Cars index. Now Cars index of powder is between Cars index of powder is if 5 to 12, then powder is a excellent flowing. If Cars index if Cars index of powder is 12 to 16, it is a good flow. If it is between 18 to 21, it is a passable. If Cars index of powder is between 23 to 35, it is a poor flow. If Cars index of powder is 33 to 38, very poor flow. If Cars index of powder is more than 40, it is very poor or cohesive powder. Cars index. Then Hausner's ratio. Hausner's ratio is again one of the another measure for flow property of powder. So if Hausner's ratio of powder is less than if Hausner's ratio of powder is between 1 to 1, if Hausner's ratio of powder is less, okay, is Hausner's ratio of, if Hausner's ratio of powder is between 1 to 1.11, then you can say powder is a excellent flowing. If Hausner's ratio is between 1.12 to 1.18, it is a good flowing, good flow. If it is between 1.19 to 1.25, fair flow, and it is possible when Hausner's ratio is between 1.26 to 1.3. और इतना याद नहीं रखना है तो फिर इतना याद रखो कितना Hausner's Hausner's ratio. If it is between 1.35 to 1.14, ये बाद में आएगा आपको, एक बाद में दो दो तीन ही वैल्यू है बाद में, उतना ही याद रखो तो चलेगा. If Hausner's ratio of powder is between 1.35 to 1.45, poor flow. If it is between 1.46 to 1.59, it is very poor flow. And if Hausner's ratio is more than 1.6, it's a very poor flow or cohesive powder. कभी कभी इसके ऊपर क्वेश्चन आता है, what is free flowing, what is आता है मोस्टली कार्स इंडेक्स के ऊपर आ चुका है या इसके हाउसनेस के ऊपर आ चुका है, एंगल ऑफ रिपोज के ऊपर भी आ चुका है. एंगल ऑफ रिपोज, what is the angle of repose? It is nothing but it is not a precise measurement of flow properties of powder, but it is a commonly used tan theta is equal to 2h upon d, 2h upon d, where h is the height of powder of pile and d is the diameter of powder. Angle of repose is nothing but the static angle which is measured by fixed plane method or fixed cone method. Static angle measured by fixed panel method or fixed cone method. ये दो मेथड होता है स्टैटिक. काइनेमेटिक और डायनामेटिक एंगल ऑफ रिपोज इज मेजर्ड बाय रोटेटिंग सिलेंडर मेथड. रोटेटिंग सिलेंडर मेथड में काइनेमेटिक और डायनामिक एंगल ऑफ रिपोज मेजर करते हैं, एंड टिल्टिंग बॉक्स मेथड इससे भी एक मेजर करते हैं. Now, if angle of repose of powder is between 25 to 30, excellent flow is there. If it is between 31 to 35, good flow is there. If angle of repose of powder is 30 to 40, 40 to 41, 40 to 41 to 45 possible. Okay, angle of repose of powder it is between 40 to 55, overflow 56 to 65, very poor flow, and more than 66, very poor flow. Particle size determination and size range analyzed. Sieving method से अपन ने already देखा है. Sieving methods you can analyze the particle size in range of 20 to 1.25 lakh. Sieving method से 5 to 120 micrometer. Sieving method photo etched sieve, photo etching sieve अगर है 5 to 120, अगर perforated sieves है तो 1000 to no 1000 to 12.5 lakh micrometer. Optical microscopy 0.5 to 150 micrometer. Electron microscopy say you can measure particle size in the range of 0.001 to 5 micrometer. Sedimentation method 1 to 50 micrometer. Centrifugal sedimentation से आता है 0.01 to 5. Culture counter method से 1 to 200 micrometer. Okay, light scattering, laser light scattering 1 to 1000 micrometer. Now compressibility and flow properties of certain pharmaceutical additives. तो मटेरियल देखो कौन सा है, सेल्यूलोज डेरिवेटिव, इसका परसेंट कंप्रेसिबिलिटी होता है 11. एन ओके, एम कंप्रेस का होता है आपका 15%, एम कंप्रेस 15% होता है. कार्स इंडेक्स दोनों भी क्या है, एक्सलेंट फ्लोइंग है. So कार्स इंडेक्स of cell type income हेलो आई एम इन लेक्चर मैं कॉल करता हूँ आपको ओके देन स्टार RX 1500 ये क्या है डायरेक्टली कंप्रेसिबल स्टार्च होता है लैक्टोज मोनोहाइड्रेट उनका कार्स इंडेक्स होता है 19 सो दे आर फेयर पासेबल देन मेज स्टार्च एंड डाई कैल्शियम फॉस्फेट DCP इसका कार्स इंडेक्स होता है 627 विच इज अ पुअर फ्लोइंग मैग्नीशियम स्टील जो होता है उसका कार्बन इंडेक्स 31 इज पुअर फ्लोइंग टाइटेनियम डाइऑक्साइड है ना उसका कार्बन इंडेक्स 34 वेरी पुअर फ्लोइंग है ना ठीक है डाई कैल्शियम फॉस्फेट डाइहाइड्रेट उसका कार्बन इंडेक्स होता है 41 एंड इज वेरी वेरी पुअर फ्लोइंग टाल्क जो होता है ना टाल्क का कार्बन इंडेक्स होता है 49 वेरी वेरी पुअर फ्लो सबसे पुअर होता है टाल्क so that is a again few examples of pharmaceutical material with their Car's index. Now solubility analysis. Solubility analysis because solubility ultimately affect the dissolution and lastly bioavailability. So focus on the drug solvent system. What is solubility? Okay. What is solubility? It is nothing but the equilibrium between solid state and solution state when there is an equilibrium between solid state of the material and solution state of the material known as a solubility. So focus on drug solvent system that occurs in a pre-formulation. Solubility that affects the bioavailability lastly. The analysis involves the determination of solubility by using HPLC, UV, GC, you know, fluorescence spectroscopy, etc. Solubility analysis में आएगा पर determination of pKa, pH, solubility profile, temperature depends, the impact of temperature on the solvent, dissolution, solubility product and solubilization mechanism. इसके अंदर ये आता है और determination of pK means क्या dissociation constant of drug pK is nothing but the dissociation constant of drug we have seen most of the drug they will either remain ionized or remain unionized depending upon the pH of surrounding medium है ना तो percentage ionization or percentage unionization that is a function of pH and pKa of drug pH of surrounding medium and pKa of drug है ना जैसे फॉर एग्जांपल अगर कोई एसिडिक ड्रग है एस्पिरिन तो एस्पिरिन एसिडिक है हाँ वो स्टमक में जाएगा स्टमक में क्या है एसिड एस्पिरिन इज अ वीक एसिड स्टमक में क्या है स्ट्रॉन्ग एसिड so it will remain unionized and it will remain unionized. Unionized form of that compound is lipid soluble. It is easily get absorbed. So aspirin get absorbed from the stomach only. Now if drug is a weak base and if it goes to the intestine, so intestine में pH होता है more than 8 है ना तो अल्कलाइन pH होता है तो अल्कलाइन pH में क्या है weak base क्या करेगा it will remain unionized and weakly basic drug they are absorbed from the intestine. Likewise, that ionization percentage ionization is a function of pH and pKa. The pH of medium and pKa of drug. Okay, unionized drug is a lipid soluble. It is absorbed easily. It can cross easily the cell membrane. Ionized drug is a not lipid soluble. It is a water soluble. There where there is a less permeation of this. The equation which is used to measure the percentage ionization is nothing but the Anderson-Selby's equation. इसके ऊपर भी क्वेश्चन आ चुका है. What is that equation? So for basic compounds, pH is equal to pKa plus ionized fraction of drug divided by unionized fraction of drug. इसको equation अपन ने already देखा है पर general pharmacology में ये. For acidic drug it is the opposite. pH is equal to pKa unionized divided by ionized. है ना या फिर आप ऐसे भी लिख सकते हैं pH minus pKa. pH minus pKa is equal to ionized divided by unionized या फिर pH minus pKa is equal to ionized divided by unionized divided by ionized. So percentage ionization is given by this formula. This is the equation 10 raised to pH minus pKa divided by 1 plus 10 raised to pH minus pKa. Now pKa determination is done by a different method. So spectroscopic method is there, potentiometric method is there, and titrimetric method is there. तीन मेथड होता है और इसके ऊपर क्वेश्चन आपको आ चुका है फॉर वीकली एसिडिक ड्रग पीके कितना होता है मोर देन 3 पीके इज मोर देन 3 whereas for weekly basic drug it is 8 to 9 and they are unionized drug these drug weekly acidic drug weekly basic drug they are they will remain unionized throughout the GIT and they are absorbed throughout the GIT. pKa determination by UV is also done. In the UV, spectral shifts they are measured and spectral shifting of lambda max. That will give you idea about the dissociation constant. It is a preferred method. Potentiometry is another method. So half wave potential is nothing but the dissociation constant. Effect of temperature on the solubility क्या होगा? Temperature बढ़ गया तो obviously solubility बढ़ता है. If there is increase in the 10 degree Celsius temperature, 5 times solubility is increased. So if there is a 10 degree change in the temperature, there is a 5-fold change in the solubility. इसके ऊपर भी क्वेश्चन आ चुका है, important.